everyone, my name is Gulnara Guluzade, a PhD candidate in animal production and food safety at the University of Bologna, Department of Veterinary Medical Sciences. Today, I'm excited to present my research project titled Implementing Next Generation Sequencing Techniques to Identify and Trace Foodborne Pathogens in Food Products and Improving the Food Safety System in Azerbaijan according to European regulation. Let's begin by looking at the broader context that inspired this research. From farm to fork principle, when we talk about the food safety, it's important to remember that every step matters from farm to fork. These principles ensure that food is monitored and controlled throughout the entire production chain, starting from the farm, continuing through processing and distribution, and ending at the consumer's table. We are focusing on raw milk and cheese production, particularly in Emilia-Romagna, Italy, within the framework of the Stalla Modello Sustainable Milk Project. We analyze raw milk and raw milk cheeses at 745 and 90 days of ripening, aiming to enhance the sustainability of their production, ensure food safety, and reduce waste while addressing the impact of climate change. This initiative is funded by the MASAF under the National Supply Chain Contract, which supports innovation and sustainability in Italian agriculture. Now that we understand the context, let's move to the main objective of the project. My project has three main objectives: to monitor and detect foodborne pathogens from dairy products in the Emilia-Romagna region of Italy using whole genome sequencing. Secondly, to use these data to understand how pathogens spread, identifying contamination sources, transmission route, and factors influencing their persistence or reduction along the food chain. Finally, to apply these insights gained in Azerbaijan by developing science-based guidelines for food safety authorities aligned with European Union standards. One Health Zoonosis Report 2023. In Europe, foodborne pathogens such as Listeria, Salmonella, and E. coli remain a major concern, especially in products of animal origin like dairy. According to the One Health Zoonosis Report 2023, outbreaks are still being detected despite strong monitoring systems which show how complex contamination routes can be. This report highlights the urgent need for more precise and faster tools to identify, trace, and control these pathogens, and this is exactly where NGS makes the difference. But how we can achieve all these? Let's take a closer look at the methods and technology used. Why next-gen sequencing? Traditionally, macrobiological methods are valuable, but they often lack the precision to trace pathogens across complex food systems. This is where NGS comes in. NGS technologies, including whole genome sequencing and shotgun metagenomics, can generate high-throughput genetic data, allowing us to distinguish between strains of the same species and to trace contamination sources along the entire food chain. By implementing VGS, we can obtain complete genetic fingerprints of pathogens, making it possible to trace contamination sources across the food chain and to prevent future outbreaks. In other words, NGS turned DNA information into a powerful tool for microbial forensics in food safety. Let's now explore how these techniques are being applied in practice. My research follows the farm-to-fork approach, starting with sample collection of dairy products in compliance with ISO standards to ensure traceability and representativeness. I was trained in microbiological analysis and the use of the Maldetov mass spectrometer, as well as the whole genome sequencing for microbial typing and characterization. The data are processed using bioinformatic tools to identify the species, resistance genes, and virulence factors, which together allow us to assess the microbiological safety of dairy products. But this project is not only about data, it's also about impact and collaboration. As part of the Giordana Masetti Research Award 2025, I had the opportunity to be a guest scientist at EFSA, the European Food Safety Authority. There, I deepened my understanding of genomic characterization of foodborne pathogens and learned to monitor strains posing potential threat to consumer health in EU level. This experience will help transfer European expertise to Azerbaijan, improving its national food safety system and ensuring better alignment with EU practices. Finally, let's look at what we expect this project to deliver. The outcomes of this research include establishing a whole genome sequencing database of foodborne pathogens from dairy products, building the scientific and technical capacity to integrate VGS data into risk assessment models, and most importantly, contributing to the harmonization of food safety regulation between Azerbaijan and the European Union. This means safer food, stronger science, and more connected international efforts to protect public health. and thank you very much for your attention.